Hello everyone. Today we will be studying about morphology and culture characteristics of Streptococcus pneumoniae. So let's come and see the microscopic picture of Streptococcus pneumoniae. How it looks in microscope. It's lancet shape. That means one end will be pointed and another one will be broad. And in case of Streptococcus pneumoniae, the broad ends are facing towards each other while the narrow end are opposite to each other. So it's a diplococcus, and as you can see in the image, there is diplococcus, broader end towards one side and narrower end to the opposite sides of each other. It's gram positive. That means it will look purplish in color since it is taking the color of crystal violet dye, which is the primary dye in gram staining. And size range will be between zero point 0.5 to 1.25 micrometers in dia. As I have already told you, it will be a diplococci, but while preparing a slide, if some pressure is given, then this diplococci may break and you can see it as a single cocci. While these are diplococci, they are covered with capsulate, and so they are called encapsulated, and encapsulated strains are usually virulent strains. Capsule. Now, how it can be cultured? The cultural requirements which it needs to grow in the media is complex nutrition. So it grows readily if the media is containing blood, serum, or sugars. That means it grows on enriched media. Even it's good on chocolate agar, and it can be grown at 37 degrees centigrade with the optimum pH 7.4 to 7.6, and it can tolerate oxygen since it is a facultative anaerobe. It can grow in absence as well as in presence of oxygen. Then how the capsule is seen? Capsule can be seen through negative staining by Indian ink. That means in a phase contrast microscope, all the capsules will show. We can see in the negative during negative staining, we can see the hollow structures which I have put as an image over here, and these are the capsules. And if you are performing differential staining, then you can use primary stain as crystal violet and counter stain, that is 20% copper sulfate. What this counter stain will be doing, it will be decolorizing and removing the crystal violet, and then you can see all the background or bacteria will be purplish in color, while the color of this will be hollow structures will be seen as the capsule. So this is how you can see the microscopic morphology of capsule. What are the medias it can grow? What are the media on which we can grow? Because media is plural and medium is singular. So it can grow on nutrient agar medium, it can grow on sheep blood agar medium, it can grow in Columbia horse blood agar medium, it can grow on crystal violet nalidisic acid media, that is gentamicin agar media, and this is the selective medium for it. Why it is selective? It's selective when it inhibits. Selective medium is the media which inhibits the growth of one bacteria and favors the growth of the bacteria which we are interested in. So if we are growing Streptococcus pneumoniae on gentamicin agar medium, then it is a selective medium for Streptococcus pneumoniae. And it can also grow in liquid media, these like glucose broth, TSB, triple sugar broth. Some broths it can grow in broth. It will give a opaque appearance. That is, then we can know that yes, this is the uh, organism of interest, that is Streptococcus pneumoniae is growing. Then you have to perform gram staining and see how it looks in microscope. If we are growing it in other medium, that is the solid medium like blood agar and all the media that I have mentioned, then on all those media it will have a circular colonies. The colonies will be circular in shape. Size range will be between zero point 0.5 to 1 millimeter. Larger colonies are observed in case of encapsulated or virulent strain. So we will be reading it in the next slide. um, Fresh colonies are dome shaped, while on prolonged incubation, colonies become flat with raised edges and having concentric rings. And this kind of colony is also called draftman or carom coin like colony. And if you will see the first picture or the image, not the picture, but yes, I have clicked it. It is a picture or image of the blood agar plate, where you can see Streptococcus pneumoniae is growing there, and the colonies are mucoid, while dome shaped, as well as greenish discoloration is there, because this is alpha hemolytic in nature. And when this colonies, this plate is aged or is incubated for a long period, on prolonged incubation, it will result like the second image, where what we are seeing, that is of the colony which is aged, and it is having carom coin like appearance, and it has become like it's called draftsman colony. So this is what I was talking, when the colony is incubated for a longer time. Now, if the strains are the ones which are highly encapsulated, so how the colony will look? The heavily encapsulated strains will be larger, will be growing. The size of the colonies will be large. It will be grayish in appearance, and they will be highly mucoid. So, in the next lectures, we will be talking about the virulent and antigenic structures of Streptococcus pneumoniae. Till then, thank you very much. Goodbye.